very much. Thank you very much for inviting me. I have to apologize for my voice. I have a cold, so it's a bit dark. Yeah, I will talk about MVA-BN, also called IMVANEX and IMVAMUNE, depending on where the product is developed or licensed, which is a new non-replicating smallpox vaccine available for updating smallpox preparedness plans. And I have also to include our usual disclaimer. So I have to go a bit back why we actually develop a new smallpox vaccine, because smallpox, as you know, has been eradicated a long time ago, but there is still Variola virus kept in approved facilities in the US and also in Russia, and it's considered one of the most dangerous pathogens that could be used as a biological weapon. So since worldwide vaccinations have discontinued more than 30 years ago, there's an increasing population that has no immunity against smallpox. So if it would be used as a biological weapon, that would be a disaster. There are remaining stocks of first-generation smallpox vaccines used during the eradication, and they have been stockpiled for emergency use by many countries. And in the US, there is since 2007 a cell-based replicating smallpox vaccine available called ACAM2000, and that is for restricted use mainly in the military. They have started many years ago to vaccinate military populations again against smallpox in the US. So that is also still a replicating product. So a few words about traditional replicating smallpox vaccines. What does it mean, replicating? So they are based on vaccinia virus strains, and they are administered using a special bifurcated needle, as you can see here, by multiple punctures. And this causes this typical vaccine take, which is the only historical measure of efficacy for a smallpox vaccine. When ACAM2000 was developed, of course, there was no smallpox around, so vaccine efficacy could only be proven based on non-inferiority of the vaccine take, because this is the historical measure, and also based on neutralizing antibody titers. These vaccines are very efficacious, and they have helped us to eradicate smallpox vaccine from the world. However, they have serious side effects, and that's the reason why we have to develop a safer vaccine for future use. There are many problems with these vaccines that are rare, but there's an increased risk for some populations, mainly the immunocompromised populations, for example, HIV-infected people, and also people with atopic dermatitis, with skin diseases. And there's also quite a high incidence of myopericarditis observed during clinical trials with ACAM2000 and also in the past. So actually this leads to contraindications for replicating smallpox vaccines in a pre-vaccination scenario. 5% of the total population cannot receive these vaccines, and this is mainly the immunocompromised and persons with eczema and persons with a history of eczema as well. So in contrast to the traditional replicating smallpox vaccines, MVA-BN is non-replicating in human cells. It's administered subcutaneously or intramuscularly, so not by vaccination by multiple puncture or by a bifurcated needle, just as a normal vaccine. And there's no vaccine take developing, and it cannot cause transmission to cell for others because it's not replicating. So it's designed for the general population, which includes immunocompromised people and people with skin diseases. However, since there's an issue with demonstrating efficacy, we had to develop a new strategy, because no vaccine take, which is the historical measure, so we had to develop another strategy to prove efficacy of this product. So it's a lot about also the animal rule developed by the US for these kind of products. So we actually started in animals to look for an equivalent immune response to conventional smallpox vaccines, and then linked this to an equivalent protection to conventional smallpox vaccines, and this again linked to the human situation in terms of immunogenicity. So to induce an equivalent immune response to conventional smallpox vaccines in people. I can only show you a few data because we have developed MVA-BN since more than 10 years, actually more than 12 years. So there's plenty of preclinical and clinical data available, and I can, in the shortness of time, show you only a few key data. These are data in mice where you can see after 1 or 2 shots of MVA-BN compared to Dryvax that you see similar total antibody titers and also neutralizing titers for these products. The same is true in monkeys, non-human primates. Here we used as a comparator ACAM2000, the new vaccine. And you see here after 1 or 2 doses of MVA-BN, you see similar titers, actually equivalent titers or even higher titers. This is the group that received 2 doses of MVA-BN. And this is the group that received only 1 dose. We gave this at day 28 to be better comparable. And as you can see, the same is true for neutralizing antibodies, equivalent immune responses after 1 or 2 doses of MVA-BN compared to the traditional smallpox vaccine. And we look at protection. This is a very good model, an acromegalia challenge model in mice. This is very much mimicking variola in humans. And you can see again, we compared 1 dose of MVA-BN, 2 doses of MVA-BN versus Dryvax, and also control animals. The control animals were all dead by day 7, while the mice that received 1 dose of MVA-BN or 1 dose of Dryvax, they had little symptoms, but they recovered completely, and the mice that received 2 doses of MVA-BN not even developed symptoms. We have also lots of data in non-human primates. We have an aerosol model, and we have also an intravenous model. This is the data from intravenous challenge model with monkeypox. You see here the placebo group, all the monkeys have died, and 1 or 2 doses of MVA-BN and also ACAM2000 has protected all the monkeys. We have also shown that the antibody responses correlate with survival, so the probability of survival, and here you see the ELISA and the PRNT data, and all these data are derived from intravenous and aerosol monkeypox challenge studies performed in non-human primates. So in the human situation, it's more difficult. That was a time when Dryvax was still licensed in the US and still available for vaccination and in clinical trials. It was a small study where we have seen, again, as in animals, 1 or 2 doses of MVA-BN given either sub-Q or intramuscular is as good as 1 dose of Dryvax. We wanted to confirm these data in a larger clinical trial. However, unfortunately, this is no longer allowed in the general population to use replicating smallpox vaccines for safety reasons. So we had actually only a chance to do a retrospective analysis. What we did, we received clinical samples from 5 NIH-sponsored studies that used Dryvax or Vetvax, and compared these to MVA-BN responses in using the same validated ELISA and PRNT assays. Actually, we repeated the sample analysis with our validated ELISA and PRNT, and this is all in healthy vaccinia-naive subjects, so people who have never seen a smallpox vaccine before. So all the people we actually included in our clinical trials have been involved in here. So, and what you can see here, again, ELISA and total antibodies and neutralizing antibodies, we have shown here that MVA-BN responses are non-inferior to the responses induced by traditional smallpox vaccines. The good thing is that we are now, as we speak, initiating a clinical trial actually against ACAM2000, so a non-inferiority trial. This is in the military setting. This is the only chance where we can actually do such a clinical trial with such a vaccine. So there will be new data coming in the phase 2 non-inferiority setting. So we have actually proven what we wanted to prove, see equivalent immune responses in mice and non-human primates. We have seen equivalent protection in mice and non-human primates, and we have also seen comparable immune responses to Dryvax in humans, and there will be comparable or non-inferiority data available when the clinical trial has been terminated, that has just been initiated. This is just to give you an overview of what has been done over the last 12 years. This is 16 clinical trials that have been completed with MVA-BN. This includes large clinical trials, phase 1 and phase 2 trials in HIV-infected subjects and in people with atopic dermatitis, so populations that cannot receive a replicating smallpox vaccine, which is very important. And the total database adds up to 3,452 subjects, and this has already doubled since then because this is the completed trials and we have 4 ongoing trials. But these data actually went into a submission package for Europe and Canada. We have already since 2010 a pre-emergency use authorization in the US for MVA-BN under the trade name IMVAMUNE. This has been based on the data we have generated in HIV-infected subjects and in atopic dermatitis subjects. And that actually triggered the delivery of 20 million doses to the US Strategic National Stockpile. So we have a big manufacturing in Denmark and have delivered already all the 20, or are in the final stage. We have delivered all the 20 million doses and actually are delivering additional doses now. And then we have received, in 2013, we have received licensure for IMVANEX. It's called IMVANEX in the EU and IMVAMUNE in Canada for this product based on the phase 1 and phase 2 data, because there is obviously an urgent need in countries where there is no ACAM 2000 licensed, which is only in the US, and there are not many first-generation smallpox vaccines left, and especially, and that is why this in Canada, they have licensed it for people that cannot receive replicating smallpox vaccines. So there is an urgent need for these populations. So that's the current status. As said, we have 4 ongoing clinical trials because we have still to do a phase 3 trial. That is actually already finished. We are in the analysis phase, and there is also 1 clinical trial done by the NIH currently. The majority of this clinical development program has been actually funded by the US government, by NIH and by BARDA. And so the database has been doubled since we have submitted for licensure in Europe and Canada. And you can see there are almost 1,000 subjects that are contraindicated to receive replicating smallpox vaccines included. Just a few words on the summary on the clinical safety. We haven't seen in all these subjects no trends or clusters for unexpected or serious adverse reactions. The vast majority are local and systemic reactions that are anyway expected with vaccines and that are mild to moderate and resolved rapidly without any intervention. And especially this has been also safe in all these risk populations. And we have also used a freeze-dried formulation. There's currently also a freeze-dried formulation under development, a new freeze-dried formulation. So we don't see any difference here in safety. And also between vaccinia naive and experienced populations, so people who actually have received the smallpox vaccine in the past that can receive safely this product. And we haven't seen any confirmed case of myopericarditis or any other cardiac inflammatory event in any of our clinical trials. So it's a very comparable safety profile to other modern licensed vaccines. So countries are updating their preparedness based on the licensure of IMVANEX now. The US ordered 4 million doses to the SNS just recently. Canada, both the Ministry of Health and the Department of National Defense ordered doses in August. And France has updated their preparedness plan following the licensure, so they have an immediately stated requirement of 250,000 doses and a secondary requirement of 4 million doses. And other countries in Europe and Asia are also in present discussion on updating their preparedness, with a special focus, of course, on the contraindicated and first-line responders. So the approval of this product has offered or offers a unique opportunity here to update current smallpox preparedness plans and to protect the people who cannot receive the other vaccines. In an emergency, they would receive it, but with huge problems to be expected. And also it can be used to protect lab workers, first-line responders, and military pre- and post-event. Just the acknowledgements, as mentioned, the majority of these clinical trials, clinical work has been funded by NIH and by BARDA. Thank you very much.
Questions?